Good morning, ladies and gentlemen. Through this video lecture, we are going to see about a very basic instrument called microscope. And this is very common, and as I told you before, a very basic instrument needs to be possessed by every microbiological laboratory. Microscope is an instrument used to observe objects which are not visualized through naked eyes, which means there are millions and millions of organisms which cannot be seen by our naked eyes. Those organisms are visualized through an instrument, and the instrument is named as microscope. So the instrument was first designed by Sir Anthony von Leeuwenhoek. Professionally, he was a lens maker. So once he was designing his lenses, he observed there are organisms which are very smaller in nature, and they are present in abundant number in rainwater sample. So which kindled him to design the instrument exclusively for observing organisms of very smaller dimension, and it makes him to design the world first microscope called a simple microscope. It was named as simple microscope because it contained only one lens. So the first designed simple microscope contained only one lens. The one what you are observing here is a compound microscope, and this is otherwise called as student microscope. So this common version of microscope is very essential for all microbiology based experiments, right? Let's see the parts in detail. And this is called eyepiece. This one is called eyepiece. So as I told you before, this is compound microscope because it contains more than one type of lens. So the one is called eyepiece. The other one at the other end is called objective piece. This is called the objective piece lens. And the eyepiece is fixed. The eyepiece is fixed, and its power is 10x. So through this piece of lens, you could magnify the object 10 times than its original size. That is why it is given as 10x power. And this area is called a hollow tube. This area is called a hollow tube. The reason is it is hollow in nature, right? The other end of the hollow tube is called objective piece, or this is otherwise called as nose piece. This is otherwise called as nose piece. There are three types of lenses in the nose piece. One is low power, second one is high power, and third one is oil immersion lens. Low power, high power, and oil immersion. The magnification power of the low power lens is 10x. The magnification power of high power lens is 45x. And the magnification power of oil immersion lens is 100x. So with the help of a normal compound microscope, the maximum times of magnification is 1000 times, which means a sample in the slide can be magnified up to 1000 times from its original image. So this is what a compound microscope can do. And this is, as I told you, this is a very basic version of microscope. Let's summarize. The one is called eyepiece, which is constant, and the power of the eyepiece is 10x. And this is hollow tube. The other end of the hollow tube contains three lenses: low power, high power, and oil immersion. Right. And this is the way to handle a microscope. While carrying your microscope in your laboratory, this is the way to carry the microscope, like carrying a duck. This is called neck portion. This is called neck portion. This is called base of the microscope. So use your left arm to bear the base of the microscope. So with your right hand hold the neck of the microscope. So this is the proper way to carry, and this is the proper way to avoid accidental loss of lenses from the microscope, right? Fine. And now we are going in detail. So this is the way, right? Use your left hand, right? Press here and uplift the portion, right? Watch me very closely. Use your left hand, press here, press at the base of the microscope, lift it gently, and this portion is called mirror. There are two types of mirror in a compound microscope. One is called concave mirror, another one is called convex mirror. And this portion is very helpful for observing the light from the light source. And a small point to recall, I told you the name of the microscope is student microscope or compound microscope. There is another name to this microscope called light microscope, because a source of illumination is required, and this mirror is used to trap light from its source. The light may be a natural light or the light may be an artificial light, right? And over the mirror there is a small portion. This is called diaphragm, right? This is called diaphragm. Diaphragm is the heart of the microscope because it controls the amount of light entering into the microscope, because entry of overexposure to light may cause blurring of image, right? You couldn't or you cannot visualize the image clearly. So light intensity is very important, and the light intensity is controlled by this unit called diaphragm, and this contains a small loop, and this portion control the amount of light entering into the microscope. So this is called diaphragm. This is otherwise called as the base of the microscope or heart of the microscope, which control the amount of light entering into the area, right? And above the diaphragm, this is called a platform for mounting your specimen, and on either side of the platform or working stage there are two clips. You could find here there are two clips which is used to hold the slide tightly, because while observing the specimen the slide should be in its place, otherwise it is very difficult to visualize the image at the right position. So to enable the slide in its place, use these clips, and there are two knobs here. One is called coarse adjustment, this is called fine adjustment knob, and this one is called coarse adjustment knob, right? So over the mechanical stage or platform there are two knobs. This one is fine adjustment knob, this one is coarse adjustment knob, right? Another important thing with respect to microscope is how to dismantle this area. See, whenever you are handling microscope, use both of your hands, and this is how you have to scroll your coarse adjustment knob, and this is how you have to scroll your fine adjustment knob. Don't use single hand, because this will definitely spoil the smoothness of the axis, right? Don't use your single hand. To dismantle this area, right, press your hand like this, or gently scroll the coarse adjustment knob, right, to the most, right. Finally, so just lift this hollow tube like this. Just lift the body tube, it will come out. And this is the main area. See, you could find the arrangement of axis. This will not be spoiled. If it is spoiled in the sense, the smoothness of the movement of the body tube will be entirely spoiled, right? So use both of your hands to adjust the fine adjustment as well as coarse adjustment. Once after taking the hollow tube in your hand, this is called the canal, hollow canal, through which the light enters into the eyepiece. And as I told you before, this one is the low power lens and this one is your high power lens, and this will be frequently cleaned with filter paper or tissue paper to remove the dirt particles, and which will help you to visualize the image clearly. So gentlemen, and this portion is called diaphragm. I told you, diaphragm is the heart of the microscope, and it controls the amount of light entering into the axis, right? There is a knob here. You can adjust the knob to control the amount of light entering into the microscope. And the one is, the thing which is inside the diaphragm is called aperture. See, the hole is very small in nature, so low amount of light alone entering into the pathway. Once you adjust this, the size of the diaphragm increases so that more amount of light pass through the diaphragm and it reaches till your eyepiece lens. So this is to control the amount of light entering into the microscope. So this is a very important portion for every microscope. So please remember, even though this is a very simple instrument, but it is a very basic one and it need to be learned by every one of us, because we have to be very strong in fundamentals. So let's recall all the things. This is the microscope, compound microscope, and this is eyepiece lens. The power of the eyepiece lens is 10x, and this area is called body tube. This is hollow in nature, which means nothing is present in between objective lens and eyepiece lens. It is hollow in nature. And the objective piece lens is otherwise called as nose piece lens, and the nose piece contains three different types of magnifying lenses: low power, high power, and oil immersion. The power of each lens is different: 10x, 45x, and 100x. So the maximum magnification power of a compound microscope is 1000x, which means what? We can magnify the bacteria up to 1000 times from its normal dimension, right? And this is the neck of the microscope, which is used to carry hold the microscope tightly, and this portion is called base portion, and this is mechanical stage. This is slide holder. This is to hold your slide in its proper position. This is coarse adjustment knob, and this is fine adjustment knob, and this one is diaphragm. So whenever you are entering into cell biology laboratory, you people are supposed to handle this basic instrument So learn it very carefully. As I told you before, we all have to be strong in fundamentals, right? Thanks for watching the video. Stay tuned with us. Thank you.
Thanks for watching our video. Stay connected with us. Subscribe our Anjak Digital channel. And don't forget to click the bell icon to get the latest updates. See you in the next video. Thank you.